Hello, my name is Zoe Wetmore, and I'm a junior here at UMass Amherst doing research in Dr. Girash's lab. And today I'm going to discuss how post-translational modifications, particularly phosphorylation, affect Hsc70 chaperone proteins, as well as my research investigating a particular phosphorylation site that is likely very important in regulating the protein's conformation. But first, what are Hsc70s, and what do they do? Hsc70s are a type of chaperone protein that help to make sure other proteins fold properly in the cell. They're absolutely crucial for cellular function as misfolded proteins cannot perform their normal functions and may in fact be harmful to the cell. Hsc70's chaperone abilities are very critical for a number of normal cellular processes, as shown on the topmost part of the diagram. Hsc70s are also critical during times of cellular stress, such as heat shock, as shown in the bottom part of the diagram. During stressful conditions, there is a greater chance for proteins to misfold and aggregate, which is very bad for the cell. Neurodegenerative diseases such as Alzheimer's, for example, are caused by aggregation of the protein tau, so it is very important that Hsc70s help to prevent such aggregation in the cell or signal for badly misfolded proteins to be degraded. I should also mention that Hsc70s are a category of protein and not one specific protein. For instance, my research revolves around Hsc70, the human constitutively expressed version of Hsc70. Another common type of human Hsc70 is HSPA1, which I'll mention later also, that are specifically upregulated in response to cellular stress. There are also Hsc70s in other species as well, including the well-studied E. coli chaperone, DNAK. Regardless of the type of Hsc70, however, they all have the same central components: a nucleotide binding domain, or NBD, a beta substrate binding domain, or beta SBD, an alpha SBD, or alpha helical lid that can close and open over the beta SBD, as well as a dynamic linker region that connects the NBD and SBD together. As their names imply, the NBD binds nucleotides, specifically ATP, and the SBD binds client proteins that need folding assistance. But how does the protein work? Now, I'm sure you can imagine that in order for Hsc70s to function properly, they first need to bind the misfolded client protein, help it to refold, and then release the client so the cycle can continue again. This process of substrate binding and release is modulated by Hsc70's allosteric cycle, as shown in this diagram. The allostery in this case is referring to ATP binding and ADP release at the NBD, which directly affects whether substrates can bind to the SBD. All right, so looking at the start of this process on the right, ATP binds to the NBD and the protein is docked since the NBD and beta SBD are interacting with each other. In the dock conformation, the alpha helical lid is swung open so new substrates can enter the SBD. Hydrolysis of ATP to ADP causes the protein to enter the undock conformation, where the NBD and beta SBD move apart and stop interacting with each other, as shown in the leftmost part of the diagram. In the undock conformation, the alpha helical lid is closed over the SBD, so the client is trapped inside, allowing Hsc70 to help the protein to fold. Release of ADP and binding of ATP allows the folded substrate to leave and the process can start again. It's also worth noting before moving on that ATP binding and hydrolysis does not happen on its own. Co-chaperone J-domain proteins are involved in mediating the ATP hydrolysis, and nucleotide exchange factors, or NEFs, are involved with exchanging the ADP for an ATP in the cell. Like I said at the outset, I am very interested in phosphorylation sites, and right now I'm particularly focusing on threonine 163 and Hsc70. Not much is known about the exact biological significance of the site, but it is located right on the interface of the NBD and SBD and likely plays a significant role in the allosteric cycle of Hsc70, as I will demonstrate in two PyMol images. First, I'm going to show you where the residue approximately is in the undock conformation, and we know this structure is the undocked conformation because the NBD and SBD are far apart and the alpha lid is occluding the beta SBD. You can also see the nucleotide binding cleft right here, and then the actual area of substrate binding right here. Note that this is a crystal structure of DNAK, since there is no full-length crystal structure of Hsc70, which is also why the highlighted yellow 163 equivalent site is arginine and not threonine. But the general structure of DNAK and Hsc70 are close enough that I can show you the general location of the T163 residue. Note that the residue is located on the NBD, but is not making any interactions with the SBD in the undock conformation. Now we're looking at the dock conformation, which we know is the case because the NBD and SBD are interacting and the alpha helical lid is flipped open away from the beta SBD. Now you can see the yellow 163 equivalent residue is right next to the alpha helical lid on the interface between the two domains. The close proximity of the residue of interest with the SBD alpha helix and DNA-K demonstrates how adding a phosphoryl group to T163 and Hsc70 could change the nature of the interactions between the NBD and SBD, since a phosphoryl group would bring negative charge and cause the threonine residue to occupy more space than it would normally. So the original hypothesis I made from those structures was that phosphorylated T163 Hsc70 would favor domain undocking compared to unphosphorylated T163 Hsc70. This hypothesis was further informed by the Main et al. 2018 PNAS paper released by this lab that found that a V163R mutation in HSPA1 caused a protein to favor the undocked state. This V163R mutation might be similar to threonine becoming phosphothreonine, since in both cases the 163 residue is increasing in size and gaining more charge, despite the fact that arginine and phosphothreonine are oppositely charged. You could further guess that phosphorylated T163 would behave similarly to the HSPA1 V163R mutation, since HSPA1 and Hsc70 have 86% amino acid sequence similarity, particularly in the areas of the NBD and SBD around the 163 residue. In order to test my hypothesis, I would need phosphorylated T163 Hsc70 proteins to compare to unphosphorylated wild-type Hsc70 proteins. In order to mimic cellular phosphorylation of T163, I have T163E Hsc70 mutants. Glutamic acid is rather similar to phosphothreonine since both are similar in size and charge, although of course they are not quite identical, so it is a decent substitute for T163 phosphorylation in vitro to begin with, but it would also be worthwhile to potentially work with actual phosphothreonine down the road once I get some baseline data. So far, I've been focusing on obtaining all the Hsc70 plasmid variants I need in lab via site-directed mutagenesis, as well as obtaining the proteins themselves by transforming the construct plasmids into Rosetta E. coli cells and inducing translation. And I've been purifying the Hsc70 proteins themselves using nickel affinity chromatography since all the proteins are His-tagged. In the future, I plan to compare my wild-type and T163E Hsc70 proteins in a few different ways to evaluate my hypothesis. First, I would want to do thermal stability tests to see if my T163E mutant is destabilized at all compared to the wild type, as well as circular dichroism, or CD, to see if there are any secondary structure changes in the mutant. I expect the mutant to not be destabilized when there really shouldn't be any perturbation in the secondary structure either, since Hsc70 shouldn't be greatly destabilized by phosphorylation of one residue that likely affects its ATPase cycle. If it is significantly destabilized, then I might need to produce a better phosphomimetic mutant or reevaluate my assumptions. Additionally, I hope to do ATP hydrolysis assays to compare my wild-type and mutant proteins. I want to check to see if there's any differences in ATP hydrolysis in the mutant because based on my hypothesis, it is possible that phosphorylated T163 causes the protein to favor the undocked state by increasing the rate of ATP hydrolysis in the NBD. I also want to perform substrate binding assays, which can give an approximation of the KD, or dissociation constant, for my wild-type and mutant proteins. The higher the KD is, the more the substrates tend to dissociate from the SBD. I would expect the KD for the mutant to be lower than the wild-type, since in the undocked state, substrates shouldn't be able to dissociate since the alpha helical lid would be occluding the beta SBD. While substrate binding assays will give me an idea of whether the protein's docked or not, I would still need to do NMR analysis of my mutant and wild-type proteins to get direct evidence of whether the protein's undocked or not, which will likely be one of the last experiments I perform. All right, before I end my presentation, I'd like to thank Dr. Girash for helping me to get involved in research and supporting my project, Dr. Clerico for being an awesome lab mentor and helping me in lab, as well as Dr. Main for his discoveries on the 163 site and HSPA1, as well as his work on Hsc70s that has been really foundational for my own project, and everyone else in the Girash lab for all their help and support. Thank you so much for watching.